We are continuing our look at the processes that form urine and adjust blood composition. So in a previous lecture, we saw how glomerular filtration forms the filtrate. Now what we will spend some time looking at is a process called tubular reabsorption. As you probably guessed, tubular reabsorption occurs in the renal tubules. So here we have a simple diagram illustrating tubule reabsorption. So just to remind you of what's going on in this simple diagram of our nephron here is that blood is flowing from the cortical radiate artery. This artery right here would be the cortical radiate artery. Blood is then flowing from that cortical radiate artery into the afferent arteriole, which is right here of the glomerulus. The glomerulus would be this ball of capillaries right here. Filtration would be created at the glomerulus here, and it would be collected by the glomerular capsule, which is here in green. And that filtrate would be sent to the renal tubules. One of the other things I want to show you here too is after blood leaves the glomerulus, is that blood leaves the glomerulus through the efferent arteriole here and proceeds down to tubules or blood vessels that pass in close proximity to the renal tubules here, and those are the peritubular capillaries if it's a cortical nephron. So selected substances in the filtrate are going to be absorbed from the renal tubule and into the peritubular capillaries. So I always kind of thought the term tubular reabsorption was confusing because to me tubular reabsorption sounds like the renal tubule is absorbing things from the blood, which is in fact the opposite matter here. Tubular reabsorption is where we're seeing selected substances in the filtrate that are in the renal tubules being reclaimed by the peritubular capillaries, which are here in close proximity to our renal tubules. So what we're talking about, or what we mean when we talk about tubular reabsorption is that tubular reabsorption is a very rapid reclamation of some of the tubule contents, and that reclamation is moving substances, selected substances, from the filtrate in the renal tubules to the peritubular capillaries that run in close proximity to those renal tubules. So we're looking at selective reabsorption of all of our organic nutrients. So this is things like glucose, amino acids, vitamins, and things like that. We're also looking at selective reabsorption of water and a number of ions. So think things like sodium and potassium when we say the term ions here. The selective reabsorption of both water and ions is very carefully controlled by several hormones, two hormones in particular here. The hormone that controls water reabsorption in the renal tubules is antidiuretic hormone, or ADH. Ions like sodium are selectively and carefully controlled, or the reabsorption of sodium in the renal tubules is very carefully controlled by the hormone aldosterone. Tubular reabsorption can be an active process. So this term active implies what? If you guessed that this term active implies that there is ATP being used, then you are correct. Active processes are where the cells in the renal tubules are using ATP to actively transport substances from the renal tubule, from the filtrate in the renal tubule to the blood, right? So we're seeing some active processes going on here. This is actually where the kidney is using most of that ATP that it requires per day is actively transporting things across the walls of the renal tubules and into the blood. This process can also be passive, and recall that passive implies that this is a process that does not require ATP. So passive processes are things like diffusion or facilitated diffusion that don't require ATP, but instead substances are following concentration gradients. We're going from high to low. The substances that are transported can take one of two routes across the wall of the renal tubule. It can take what's called a transcellular route or a paracellular route. So what those two terms imply here, and looking at this diagram, just to orient you here, on the left side, we're seeing that this is the lumen of the renal tubule here. So the material that you're seeing here is the filtrate that is making its way through the renal tubules. What you're looking at here are the microvilli on the apical surface of the tubule cells here. Remember that the apical surface always faces an inner lumen or an external surface here. So in this case, this apical surface here is the surface that faces the lumen of our kidney tubule and is the surface across which things are being absorbed from the filtrate into the blood. Over here on the opposite side, we have what's called the basolateral membrane, and this is the membrane. The membrane side of the tubule cells that is in close proximity to our paratubular capillaries here. So we have an apical membrane of our tubule cells that faces the filtrate in the lumen of the tubule, and a basolateral membrane that faces of these tubule cells that faces the paratubular capillaries here. So now that we know we have familiarized ourselves with the two surfaces of these tubule cells, we can talk about transcellular and paracellular routes. Transcellular involves movement across the plasma membrane here of the cells lining the tubules. And that also implies that transport of solutes in water is making its way through the cytosol or the cytoplasm. Remember, the cytosol is the term that we use for the fluid inside of our cells here. So the transcellular route involves moving across the plasma membrane through the cytosol, the tubule cell, out into the tissue fluid, the space between the cell and the blood vessel here, and then finally into the paratubular capillaries to be reclaimed by the blood. Paracellular involves movement between cells that are lining the tubules, not through them, but between two adjacent cells here. When we do have tight junctions and tight in the kidney tubules here, and normally tight junctions seal the space between cells so that movement of materials is not possible between two adjacent cells where tight junctions are present. But the tight junctions in the renal tubules are quite leaky compared to tight junctions in other places, like the stomach or the small intestine. The tight junctions in the tubule cells, particularly in the proximal convoluted tubule, the tight junctions here are fairly leaky, and so it allows substances like water and small solutes to easily squeeze their way between two adjacent tubule cells here and make their way into the blood. So there we have transcellular and paracellular routes of water and solute movement from the filtrate into the paratubular capillaries there. Let's look at how specific substances make their way across the tubule cell membranes or between the tubule cell membranes and into the blood here. So let's focus on sodium transport for a second. Sodium transport is important to discuss because sodium is the most abundant cation or positively charged ion that is in the filtrate. And what we see is that we'll start at the basolateral membrane. So we're going to start at the membrane of the tubule cells that's closest to the paratubular capillaries here. And so we see at the basolateral membrane here that sodium is moved out of the tubule cells by the sodium-potassium pumps. And sodium-potassium pumps are a form of primary active transport, so they use ATP to pump sodium out of the cells. And once sodium is pumped out of the cells into the tissue fluid space, it is then picked up by the paratubular capillaries. So looking at a diagram of this once again, we see that the sodium-potassium pumps, I'll go ahead and do that. I'll go ahead and circle this in green here for you. The sodium-potassium pumps are located at the basolateral membrane, and so it's promoting the pumping of sodium out of the tubule cells and into the paratubular capillaries here. So recall that sodium-potassium pumps work by what I like to kind of describe as the 3-2-1 method. We get 3 sodium pumped out of our tubule cells, 2 potassium pumped into our tubule cells here, and the cost of this movement is 1 ATP. So we're seeing ATP being involved in the movement of both sodium and potassium here. Okay, so we've seen how it moves out of the tubule cells and into the paratubular capillaries. How does sodium get into the tubule cells to begin with? That is, how does sodium get from the filtrate into the tubule cells? Let's take a look at that now. Sodium is moved across the apical membrane. Right? Remember, the apical membrane is the membrane of the tubule cell membrane that faces the lumen of our renal tubule. So it's the membrane that's facing the filtrate, or the membrane that's filtrate is being absorbed across. So we have sodium movement across the apical membrane by facilitated diffusion. And the reason why this happens, so if we go back and circle this here, so we see that there are protein carriers in the apical membranes of our tubule cells, and those are located right here. And we see that here, this solid blue arrow is indicating that sodium transport is by passive transport or diffusion. And the reason why sodium is diffusing passively from the filtrate into the tubule cell is because of the action of our sodium-potassium pump over on the basolateral membrane here. So we've got sodium being pumped out of our tubule cells here, which creates a low concentration gradient of sodium inside of our tubule cells here, because sodium is actively being pumped out and into the paratubular capillaries. So we have a relatively low concentration of sodium inside of our tubule cells, which is going to promote the diffusion of sodium from the filtrate into the tubule cell to begin with, and then the sodium-potassium pumps go to work to pump that sodium out and into the paratubular capillaries here. So we see that the action of the sodium-potassium pump promotes the passive transport of sodium into our tubule cells here. We also see that these carriers also bring in friends with sodium. So as sodium is moved in by passive transport, other substances are moved in by what's called secondary active transport. So they're essentially, secondary active transport is making use of the energy and effort spent by the sodium-potassium pumps here. So things like glucose, small amino acids, ions, and vitamins here are moving into the tubule cells from the filtrate into the tubule cells by secondary active transport, and sodium is assisting these protein carriers in moving those the glucose, vitamins, amino acids, and things like that by secondary active transport. So that's pretty cool, and it's essentially piggybacking off of that energy spending process that we see from the sodium-potassium pumps. So as I mentioned there, just to kind of summarize, primary active transport of sodium drives secondary active transport of solutes like glucose. So we see a lot of glucose being very quickly reabsorbed within the tubules here, because it does leave the blood vessels in the filtrate, so we've got to reclaim glucose. We see amino acids, which are smaller than proteins, so they can and are often found in the filtrate. Vitamins, of course, which we saw vitamins in the previous chapter, are important coenzymes, and a number of ions as well. So we've looked at sodium transport. Let's discuss water transport. So water transport is very carefully coupled to the movement of sodium, because in earlier lectures I had described water and sodium as BFFs. So wherever sodium goes, water tends to follow it. So the sodium movement into and out of the tubule cells creates an osmotic gradient for water, which means as sodium is moving into from the filtrate into the tubule cells, that's going to create this osmotic pulling force for water to also move from the filtrate into the tubule cells and then finally into the paratubular capillaries there. So therefore, water is primarily moved by osmosis. So this is a passive transport process because, again, water, like those other substances, is piggybacking off of the movement of sodium. Water transport uses a special type of facilitated diffusion channel called an aquaporin. So if we return back to our diagram here, we saw the active transport of sodium and potassium across the basolateral membrane. Across the apical membrane, we saw the diffusion of things like sodium and secondary active transport of glucose amino acids and things like that. If we turn our attention to water here, we see that these little channels right here in the magenta color are our aquaporins. And so these are the protein channels that are allowing water to diffuse across both the apical membrane, so from the filtrate in the lumen, across the apical membrane, into the tubule cells here, across the basolateral membrane, and into the peritubular capillaries here. So the presence of aquaporins is going to be very important for allowing water to move from the filtrate across the tubular cells and into the peritubular capillaries here. So aquaporins are always present in the proximal convoluted tubule. So right as filtrate is leaving the glomerular capsule and entering the proximal convoluted tubule, water is already being reabsorbed in the proximal convoluted tubule. Aquaporins can also be manufactured and inserted into the collecting ducts if the hormone, antidiuretic hormone, is present. So normally aquaporins are not present in high quantities in the collecting ducts, but as the levels of antidiuretic hormone in the blood go up, the numbers of aquaporins in the collecting ducts goes up as well. And so we see that antidiuretic hormone is an important hormone that comes into play when we're dehydrated. So by inserting aquaporins into the collecting ducts, we can highly concentrate our urine, produce a very highly concentrated but small volume of urine, and that will help us conserve a lot of body water. So let's look at where reabsorption is taking place in different regions of the tubules. So if we think about the proximal convoluted tubule for a second, we see that most reabsorption is occurring in the proximal convoluted tubule. So this is essentially the small intestine of the kidney here. So remember that the small intestine is where the majority of water absorption and nutrient absorption is occurring in the digestive system. The proximal convoluted tubule is the same way. So the proximal convoluted tubule is where a lot of the work of reabsorbing solutes and water is taking place. So exactly what is absorbed here? We see roughly just slightly more than half of the filtrate, about 65% of the filtrate volume is reabsorbed here. So that remaining 35% is reabsorbed by nephron loops or distal convoluted tubules or collecting ducts. So a good majority of the filtrate volume is reabsorbed in the proximal convoluted tubule. We see that the proximal convoluted tubule reabsorbs all nutrients, so this is glucose, amino acids, vitamins, and things like that. We also see a lot of sodium absorption here and a lot of water absorption taking place here, except in the cases of dehydration. When we're dehydrated, water will also, there's still a lot of water being absorbed here, so most of the filtrate volume is still collected at the proximal convoluted tubule, even when we're dehydrated. But we also see water absorption taking place in other regions like the collecting ducts when dehydration is taking place. So just to summarize that here in this diagram from the book, this would be the renal, this would be the Bowman's capsule here, or the glomerular capsule right here, and this would be the proximal convoluted tubule. And just kind of summarizing what we saw earlier, 65% of the filtrate volume is reabsorbed by the proximal convoluted tubule. So we see a lot of water being reabsorbed here, sodium, bicarbonate, many other ions, as well as our nutrients like glucose, amino acids, and vitamins. So, and that summarizes all of that here in this table as well, except here it's not only showing you the substances that are reabsorbed, but it's showing you their mechanism as well. So we saw that sodium potassium pumps, which are a primary active transport process, are working at the basolateral membrane to pump sodium from the cells of the proximal convoluted tubule into the peritubular capillaries there. nutrients like glucose, amino acids, and vitamins are moving across the apical membrane by secondary active transport. Water is being reabsorbed by osmosis because it's following sodium here. So let's take a look at what's going on now in the nephron loop as far as tubular reabsorption is taking place. So remember that the nephron loop is a U-shaped structure, and it has two limbs to it. It has a descending limb. The descending limb comes off of the proximal convoluted tubule and bends in its U shape at the bottom of it. The ascending limb makes its way up to the distal convoluted tubule. So in the descending limb, we see that water can easily leave the filtrate, but sodium is not able to leave the filtrate. So water can leave the filtrate because the descending limb is permeable to water, but it is not permeable to sodium. In the ascending limb, the opposite is true, where we see that sodium can easily leave the filtrate. That is, the ascending limb is permeable to sodium, but it is not permeable to water. So the ascending limb, water is not allowed to leave the filtrate. Now, this may seem like an unusual configuration, but when we discuss how really concentrated urine is formed, we're going to see that the nephron loop and the different regions of the nephron loop and their permeabilities are really important in that process. So this may seem like initially kind of an unusual configuration, but we'll see why it's important later on. So the thin segment here of the descending limb is permeable to water. The thick segment, the ascending limb here, particularly in the thick segment here, is permeable to sodium and potassium and things like that, but it is not permeable to water. So, and like I said, we'll see why it's important that sodium is allowed to leave the filtrate here but not here, and the opposite is true for water. Why is water allowed to leave here but not here? So we'll see what happens there later on. So just to summarize there, the nephron loop here is the descending limb of the nephron loop is permeable to water, and water leaves by osmosis. So again, that's a diffusion process here. Sodium, potassium, chloride, and things like that are being moved primarily by secondary active transport across the walls of the ascending limb there. So we see ATP energy being used indirectly in this fashion to move things like sodium chloride and potassium here. So finally, let's see what's going on in the distal convoluted tubule, which is the end of our renal tubule, and then also in the collecting duct, which receives filtrate from the distal convoluted tubule here. We see that in both the distal convoluted tubule and the collecting duct, reabsorption is regulated primarily by hormonal controls. And the three hormones that come into play in this process, two of which we've seen already. The first hormone is antidiuretic hormone. The second hormone is aldosterone, and we've talked about these two hormones already. We've talked about atrial natriuretic peptide in previous chapters, but we'll review its function here because it's been a little while. So let's take a look at the function of antidiuretic hormone, or ADH, in the movement of water. So recall that antidiuretic hormone, being a hormone, is released by an endocrine gland. It just so happens that this endocrine gland is located in near the brain. So it's located, antidiuretic hormone is released by the posterior pituitary gland, which is at the base of the brain. Antidiuretic hormone works to insert aquaporins into the principal cells of the collecting duct. So the more antidiuretic hormone that's present, the more aquaporins get inserted in this collecting duct, and the greater water absorption we see taking place. So as I mentioned earlier, we see antidiuretic hormone being released. Normally it's released in very low quantities, even when we're not dehydrated, but as we become dehydrated, we see the levels of antidiuretic hormone in the blood slowly start to rise, and so that helps to reclaim a lot of water at the collecting ducts for us. So recall that the principal cells are the most common of the collecting duct cells, and they have very short microvilli, and they're important in maintaining both water and sodium balance. So let's look at the effect of aldosterone. So recall that aldosterone works by affecting sodium reabsorption. So aldosterone controls sodium reabsorption. What aldosterone does is that aldosterone is, first I should mention where it's released from. Aldosterone is primarily released from the adrenal gland. And when it is released, it promotes the formation or the synthesis of sodium-potassium pumps in both the distal convoluted tubule and the principal cells of the collecting duct. So the more aldosterone that's being released from the adrenal glands here, the greater aldosterone release, the greater the sodium reabsorption we have, because we see more sodium-potassium pumps being created here. But remember the work of the sodium-potassium pump. So when sodium-potassium pumps move sodium out of the tubule cells and into the peritubular capillaries, we get movement of sodium into the tubule cells here, because sodium and potassium move in opposite directions when they're pumped by the sodium-potassium pump. So what we're seeing is that increases in aldosterone do help promote the reabsorption of sodium from the filtrate into the blood. But we see that aldosterone is going to reduce the amount of potassium that's making its way to the blood, because it's pulling potassium back into the tubule cells. Finally, we'll take a look at what atrial natriuretic peptide is, or we'll review what atrial natriuretic peptide does in terms of its hormonal effects. So remember that atrial natriuretic peptide is made by the cells in the atria of the heart. And atrial natriuretic peptide is released when blood pressure or when blood volume are elevated, because atrial natriuretic peptide influences sodium reabsorption, but in the opposite way that aldosterone does. So when we have an increase in the release of atrial natriuretic peptide, we get a drop in blood sodium. So that is, more atrial natriuretic peptide helps to essentially keep sodium in the filtrate, and sodium therefore will not be reabsorbed, and more sodium will be lost to the urine when we have high atrial natriuretic peptide. The reason for this is because remember that water always follows sodium. So if we are losing sodium to the urine, we're also losing water to the urine, and that means that there's going to be less water going into our blood, which will help lower our blood pressure and lower our blood volume when both of those variables are elevated here. So to summarize here, we saw that ADH, or antidiuretic hormone, promotes the reabsorption of water in the collecting duct here. So we see that highlighted in this diagram here. Aldosterone promotes sodium uptake in the distal convoluted tubule here. So sodium is, the reuptake of sodium is very carefully controlled by aldosterone, and it's also controlled, aldosterone also controls the reuptake of sodium in the collecting ducts as well. Atrial natriuretic peptide inhibits sodium uptake in the ascending limb of the nephron loop and in the collecting duct here. So if our blood volume and blood pressure are too high, sodium and water are going to stay in the filtrate, and those will be lost in the urine. So that will help reduce our blood pressure and blood volume. And again, just to summarize here, in the distal convoluted tubule and the collecting duct, the substances that are lost here and the mechanisms that they're lost by, or the mechanisms, I should say, that they're controlled by. So maybe I'll say the substances that are reabsorbed are highlighted in this table here, and the mechanism by which they're reabsorbed is also highlighted here as well. So recall that sodium transport across the distal convoluted tubule is a primary active transport process, and it's aldosterone regulated. Sodium transport across the collecting duct is also dependent on aldosterone, and it's an active transport process that requires those sodium-potassium pumps. Water is also reabsorbed at the collecting duct as well. It is a process, the reabsorption of water is a process that's carefully controlled by antidiuretic hormone.